name is Noriza. I will present about thermogravimetric analysis TGA. First introduction. As we know, thermogravimetric analysis TGA is an excellent thermal analysis method for measuring the composition, purity, decomposition reaction, applied temperature during process, and measure content of the intended material. TGA is also used to enhance the characteristic of materials for various applications, including pharmaceuticals, food, petrochemicals in the environment. We can also determine sample maximum temperature range by hitting it a control rate within a specific environment such as air and oxygen. The sample in TGA will show how the materials have been subjected to the thermal stress, and the thermal process will be displayed in a graph. Moving to the process, during the heating process TGA, some of the main steps that take place include the materials per sample is heated slowly at a controlled heating rate. This heating rate is important to maintain stable condition and avoid rapid temperature change, which can affect the measurement result. And second, the temperature of the sample is continuously monitoring during the heating process. This information is important to record important points such as the point of the decomposition and combustion that can occur at a certain temperature. And third, the sample which changes during heating is recorded by the TGA equipment either automatically or manually. This transformation take place as a result of the materials undergoing combustion, thermal breakdown, or volatile loss is respond to the temperature rise. And next, a TGA curve is used to calculate and show weight change data. This graph illustrates the link between the sample temperature, y-axis, and percentage change in weight, y-axis, describing the sample's burning or decomposition process. And last step, the TGA curve finding examined to access the material thermal characteristic when the data get during explanation. Decomposition point identify thermal stability, weight change, pattern, and the kind of chemical process that take place during heating all included in this. The purpose of TGA heating process is to deliver precise and comprehensive data regarding to the thermal response of the material temperature, which can ascertain for materials development, quality control, and industrial application. The video above is how we do the TGA well in the lab. First of all, use analytical balance to weigh the alumina crucible precisely. To find a combined mass of the sample and crucible, place the sample into the alumina crucible and weigh it once more. The mass of the sample should range from 2 until 5 mg. Place the sample containing alumina crucible carefully within the TGA instrument sample holder. Ensure the sensor is installed correctly and the sample holder is positioned correctly in the boulder. To get ready for analysis, shut down the furnace. Decide to heat the area at 15 degrees Celsius each minute. Set the nitrogen gas flow rate to 25 ml per minute to create an inert environment. Decide on the experiment temperature range, which should start at 25 degrees Celsius and reach almost 8,000 degrees Celsius. TGA analysis software on a computer will plot a graph showing the change in sample mass as a function of temperature. This graph is used as a data. Here the result of TGA analysis of the graph and the related concepts. First of all, decomposition point. On the TGA graph, the decomposition point is shown by two main peaks at the temperature around 285.27 degrees Celsius and 422.96 degrees Celsius. This indicates two stages of combustion or decomposition of the materials where the material undergoes significant weight loss. Secondly, thermal stability. The temperature range is which the material remains stable is not significantly affected by degradation or chemical change can be observed between the main peak on the TGA graph or the place outside the significant level of the decomposition. And composition of materials, the weight loss pattern at a specific temperature is displayed on the TGA graph, which offers information about the material composition at 350.52 degrees Celsius. For instance, weight loss of 25.06% indicates that parts of the organic stuff is in the wax start to break down at this temperature. Next, decomposition reaction. The weight change shown on the TGA graph reflects the kind of chemical reaction that makes take place during the breakdown of a substance such as combustion or thermal decomposition. For instance, gases or light product may originate in the sample or emerge at specific temperature. In addition, purity. TGA graph can be used to evaluate the purity of materials by monitoring the pattern of weight change during the heating. This residue left after the decomposition stage giving us an idea how pure the material is. Last but not least, moisture content. Although no evidence in the graphic, the material's moisture content might influence the way it composition or burns, which can impact the thermal analysis finding. Understanding the thermal properties of materials such as stability, reactivity, and composition is made easier with the use of TGA graphic data. This attributes crucial for a variety of application in both industry and research.
The next slide is detailing application. Point number 1 is material characterization. TGA is used to characterize a wide range of material including polymer, metals, ceramic and composite. It helps determine thermal stability, composition and degradation temperature. The second point is quality control. TGA is used for quality control in industrial setting to ensure material meets specified standard. Case include analyzing the content of additive, fillers and moisture. Number 3 is research and development. Scientists and engineers use TGA to develop new materials with desired thermal properties. It assists in studying the kinetic of thermal decomposition and understanding material behavior at high temperature. Next point is environmental analysis. TGA can analyze the thermal behavior of waste materials, aiding in the development of waste treatment processes. The last point is pharmaceutical. TGA is used in the pharmaceutical industry to study the stability and composition of drug formulation. Balance function measure the weight of the sample with high precision, typically in the microgram range. The importance ensure that very small weight changes can be detected and recorded accurately. The second point is furnace. The function of furnace is heat the sample at the control rate and can reach very high temperature over 1000 degrees Celsius. The important is allow precise heating program including gradual heating, isothermal hold and cooling. Next, temperature control system. The function is continuously control and monitors the sample temperature throughout the experiment. The important is provide accurate temperature control for reliable analysis. And the last one is gas flow system. The function is control the atmosphere around the sample by introducing desired gases such as nitrogen, argon or oxygen. The importance is provide a controlled atmosphere to either prevent oxidation or promote a specific reaction during analysis. For the conclusion is, TGA is essential for studying how material respond to heat. It measures weight changes as material are heat or cool, giving insight into their composition and how they break down. TGA is used widely in research, industry and environmental studies to develop new materials, ensure quality and manage waste effectively. Its precise measurement helps scientists and engineers innovate and improve product while meeting strict standards for performance and safety.
The infrared spectrum of absorption, emission, or photoconductivity can be found using Ftir. It is possible to analyze gas, liquid, and solid with this technology. The unique frequencies detected by Ftir are associated with the vibration of chemical bond in molecule. This vibration aids in the identification and measurement of the many constituents that make up a sample. Ftir spectroscopy is used in chemistry to analyze chemical compounds and investigate their molecular structure. By examining the unique frequency of chemical bond vibration, Ftir provide detailed information about the molecular makeup and functional group in a sample. Ftir spectroscopy is used in material science to characterize composite, coating and polymer. Research may learn more about this material structure, characteristic and chemical made up by examining their infrared spectra, which aid in comprehending their capabilities. First, the apparatus was calibrated according to the manufacturer instruction. Then, it was turned on and allowed to warm up for at least 30 minutes to ensure stable operation. Second, solid sample were ground into a fine powder and placed in the sample holder. Liquid sample were added as a few drops to the sample holder. Gas sample were contained using a gas cell. Third, the spectrometer produced an interferogram which the computer program convert into an infrared spectrum. The spectrum data was then stored for later analysis. Next, the spectrum was analyzed to identify characteristic peaks. Wave number were compared with no different spectra to determine functional group and chemical bonds. Next, after each sample, the sample holder was cleaned thoroughly. The workstation was also cleaned to maintain cleanliness and safety. Finally, the Ftir spectrometer was shut down.
First kali yang kita buat, kita akan push dulu instrumen ni dengan gas nitrogen. Gas nitrogen kita push dulu. Push tu sebab apa? Sebab kita nak buang moisture yang ada dalam kita punya treatment lah. Yang ni kita spektrum photometer, mana nama dia Isaac Tag, collect Isaac Tag. Yang ni collect IM Tag. Yang ni microscopy. Yang ni hanya boleh tengok dengan mata yang sampel yang tak boleh tengok dengan mata kasar. Mana dia 20 mikron yang ke bawah, 200 mikron ke bawah. Yang ni yang sampel yang boleh tengok dengan mata kasar. Yang besar-besar lah. Okay. Sampel yang kita guna biasanya kita akan guna liquid sample, boleh powder solid pun boleh. Biasa yang student makan yang extraction daun ke tumbuhan ke apa ke yang di dalam bentuk itu ke macam. Yang ni kita punya ni simple. Yang saya cakap dia push dengan liquid nitrogen dengan push dengan gas nitrogen. Push tu kita nak pastikan dia ada indicator dia. Di mana indicator ni akan jadi pink. Sebab itulah kita perlukan push. Yang ni dah kita review sebab kita dah push dalam masa 10 minit. Okay. Sebelum kita push tu, kita tukar dengan desikan dia. Desikan dia ada dalam ni. Yang lebih macam ni lah, dia lebih kurang macam si gajah lah. Lebih kurang dalam kan dia sampai gajah. Jadi kita buat 10 minit, pada pink akan bertukar daripada diri. Kerana instrumen ni dia daripada masjid dan kurang sekadar undesired carbon dioxide, water vapor apa semua dia lebih kurang seksual. Untuk first, yang ni stage dia ya, stage untuk kita letak samples. Stage dia ni, dalam ni ada diamond. Dia panggil accessories dia APR. Di dalamnya ada diamond, kita akan letak sampel dekat sini ni. Untuk first kita akan cuci dulu dengan alkohol 70%. Kenapa alkohol? Sebabnya alkohol ni dia mudah meruapkan. Jadi dia tak tinggalkan apa-apa kesan dekat sini. Kalau air, dia tinggalkan kesan. Untuk sampel, kalau liquid kita tak pakai yang ni sebab dia boleh tembus, dia boleh baca lah. Kalau untuk powder, sampel dan solid, kita kena letak benda ni. Untuk ditekan sebab dia baca daripada bawah. Dia akan rapatkan kita punya sampel dengan ni lah, kita punya detector lah.
For the functional, in FTIR spectroscopy, the critical component is where infrared light interacts with the sample, leading to molecule vibration that absorb specific frequencies. The interaction produces a unique molecule fingerprint used for material identification. The infrared microscope IN10 integrated both FTIR and microscopic function, allowing seamless transition from sample observation to spectral analysis. The kiosk software Omni collect a raw spectral data during FTIR measurement recording, the intensity of infrared light absorbed or transmitted by the sample. So about the result, we have graph which is represent an infrared IR spectrum showing peaks and specific wave number corresponding to molecule vibration within the sample. Researchers use the data to identify functional group and molecule bonds. The axis X represent wave number and the Y indicated the intensity of the signal absorbed or transmitted. Different color line represent a various data set adding the material identified and comparison. So about the result, we have graph which is represent an infrared IR spectrum showing peaks and specific wave number corresponding to molecule vibration within the sample. Researchers use the data to identify functional group and molecule bonds. The axis X represent wave number and the Y indicated the intensity of the signal absorbed or transmitted. Different color line represent a various data set adding the material identified and comparison. So about the result, we have graph which is represent an infrared IR spectrum showing peaks and specific wave number corresponding to molecule vibration within the sample. Researchers use the data to identify functional group and molecule bonds. The axis X represent wave number and the Y indicated the intensity of the signal absorbed or transmitted. Different color line represent a various data set adding the material identified and comparison. So about the result, we have graph which is represent an infrared IR spectrum showing peaks and specific wave number corresponding to molecule vibration within the sample. Researchers use the data to identify functional group and molecule bonds. The axis X represent wave number and the Y indicated the intensity of the signal absorbed or transmitted. Different color line represent a various data set adding the material identified and comparison. So about the result, we have graph which is represent an infrared IR spectrum showing peaks and specific wave number corresponding to molecule vibration within the sample. Researchers use the data to identify functional group and molecule bonds. The axis X represent wave number and the Y indicated the intensity of the signal absorbed or transmitted. Different color line represent a various data set adding the material identified and comparison. So about the result, we have graph which is represent an infrared IR spectrum showing peaks and specific wave number corresponding to molecule vibration within the sample. Researchers use the data to identify functional group and molecule bonds. The axis X represent wave number and the Y indicated the intensity of the signal absorbed or transmitted. Different color line represent a various data set adding the material identified and comparison. So about the result, we have graph which is represent an infrared IR spectrum showing peaks and specific wave number corresponding to molecule vibration within the sample. Researchers use the data to identify functional group and molecule bonds. The axis X represent wave number and the Y indicated the intensity of the signal absorbed or transmitted. Different color line represent a various data set adding the material identified and comparison. So about the result, we have graph which is represent an infrared IR spectrum showing peaks and specific wave number corresponding to molecule vibration within the sample. Researchers use the data to identify functional group and molecule bonds. The axis X represent wave number and the Y indicated the intensity of the signal absorbed or transmitted. Different color line represent a So for application, FTIR spectroscopy is a versatile technique and used across various fields. It aids in environmental analysis by detecting pollutant and assessing water and soil quality. In the food industry, FTIR ensures safety, analysis nutrient and authenticated beverage. For medical and pharmacists, it characterizes a drug, study biomolecule, and ensure quality control. In polymer science, FTIR identify polymer and assess material compatibility.
In both science and business, FTIR is a flexible analytical technique that is quite popular. Given its speedy and price, molecular identification and structural analysis, it is vital for research, quality control and environmental monitoring because it is non-destructive a wide range of material, including biological, tissue, chemical, polymer and environmental contaminants may be thoroughly examined. FTIR is a versatile analytical method used in science and industry for accurate molecular identification and structural analysis. It's essential for research, quality assurance, and environmental monitoring, offering non-destructive examination of material like biological, tissue, chemical, polymer, and environmental toxin.